Hempstead Middle School. This is Mr. Murgana and this is
Mr. G.
And we're back at it with another Mad Science Fun Friday. Guess what? Today we're going to be extracting strawberry DNA. Mr. G, do you know what DNA is?
Yes, DNA is the molecule that contains instructions for all life. So anything that's living has DNA inside of itself.
Perfect. So, Mr. G is basically saying that this strawberry is coming from a plant that's living. So guess what, Gators? This strawberry is going to have DNA inside of it. Now, the first thing that we're going to be doing is we're going to be making something called an extraction buffer. And now that's just a big word for solution. So the first thing I'm going to do is I'm going to make sure I have all my materials with me. I have my measuring cup, my dish soap, my salt, my glass cup that Mr. G is going to fill up with water, right? And also an empty glass cup just in case I mess up for right now. All right, let me move these strawberries to the side. And first thing I'm going to do is I'm going to measure out 90 milliliters of water using my measuring cup. So 90 should be about there. I'm going to stop it. I'm going to pour it into my plastic container. The next thing that I'm going to do is I'm going to measure a 1/4 teaspoon of salt. All right, I'm going to pour it in there. And I'm just going to mix it until there's no more salt in my solution. So basically, I'm mixing it until all the salt dissolves. The next thing I'm going to do is I'm going to pour 2 teaspoons of dish soap into my extraction buffer. 1 and 2. Now, I'm just going to mix it until all the soap leaves the bottom and I start seeing bubbles in my extraction buffer. All right, so I'm gonna mix it for 2 more seconds. Now, our extraction buffer is complete. You're probably wondering, why are we making an extraction buffer? Well, Mr. G, tell our Gators why.
All right, so the whole point of this experiment is to try to get the DNA out of the strawberry. But strawberries have cell walls. And so those cells, those walls need to be broken down, and the buffer is going to do just that. So what we're going to do here is we're going to take our plastic bag and we're going to take our strawberry. And what I'm going to do is remove this green part, the stem of the strawberry, and it's going to look like that. And what you're going to do is you're going to take that strawberry and place it into the plastic bag. Then what you're going to do is you're going to pour some of that extraction buffer into the plastic bag as well. You don't have to pour the whole thing in. All you're going to do is pour enough so that it almost submerges half of the strawberry. All right, so you can see that right here. Now, what you're going to do is you're going to seal the bag, close it real tight so that nothing can get out of it, and try to get all the air bubbles out of it too. Now, what you're going to do is you're just going to take your hand and you're going to squeeze the strawberry and you're going to crush it so that it basically starts to dissolve into that extraction buffer. And what's going to happen is all the solid parts of the strawberry are going to get very small. And they're going to mix really well with the extraction buffer. And right now what's happening is that buffer is breaking down the cell walls inside of that strawberry so that we can get the DNA out. You might have to do this for like a minute or two. I'm only going to do it for a few seconds here so that we can get to the next step.
So what we're going to be doing is we're going to be taking our empty glass. We're going to be putting our strainer on top of the glass. And Mr. G is going to pour this extraction buffer into this new cup.
All right, be very careful to make sure that you are pouring it into the cup. It's very easy to miss it. All of that solid material is going to be stuck in the strainer, and then the liquid stuff that we're going to use should be in the cup below.
Awesome. I'm just going to press just a little bit on the strawberries to get all that liquid out. Perfect. Now, listen up, Gators. The next thing that we're going to be doing is we're going to be pouring around 3 to 4 teaspoons of rubbing alcohol into our mixture. Now, the first thing that I want you to remember is that this rubbing alcohol should have been chilling in your freezer for at least 1.5 to 2 hours. All right. So I'm going to open my rubbing alcohol and I'm just going to pour 3 to 4 teaspoons. All right. Perfect. And guess what? I'm going to wait around 2 to 3 minutes for a white substance, a white precipitate, to be forming at the top of the liquid. All right, we'll be back with you in 2 to 3 minutes. All right, Gators. All right, Homestead Middle School, we're back at it, me and Mr. G. And right now, you should be seeing a white film precipitate at the top of this solution. And that's gonna be our DNA. Let's watch Mr. G extract this out with a spoon.
All right, so here I'm gonna take some of that DNA, and you can see some of it's on the side of the cup right here, and that white stuff that's on the spoon right there, that is DNA that has been extracted from all of the cells.
So you're telling me, Mr. G, that white stuff is gonna be what controls life for strawberries and plants?
Yeah, exactly. That's DNA, and that's found in every single one of the cells of a living organism.
All right, Gators, that's how we extracted strawberry DNA. You know the drill. If you do this at home, make sure to tag Homestead Middle School, all right? And Mr. Mutaganda will give you credit for it in the grade book. Mr. G, you got anything to add?
I just look forward to seeing you all next week for the next Science Friday.
All right, have a great Mad Science Fun Friday, Gators. See you next week.